Simplify. Don't forget to subscribe. Physiological anatomy of the gastrointestinal wall from Guyton and Hall. This is our digestive tract, which consists of the mouth, pharynx, esophagus, stomach, small intestine, large intestine, rectum, and anus. The digestive process begins with the ingestion of food. The food that you have taken into your mouth is mechanically digested when you chew it. Then you swallow it, which is the propulsion of the food from the oropharynx to the esophagus, where its peristaltic movements begin, which are the contraction and relaxation of the smooth muscle wall, which helps to get the food further forward. This food is then moved to the stomach, where it is further mechanically digested by churning and mixing process. Then this food is moved to the small intestine, where it is chemically digested with the help of enzymes. These enzymes convert your macromolecules into the micromolecules, which are then able to be absorbed into the blood vessels and lymph vessels, from where they are transported to all the tissues of the body wherever they are required, and the food that was unable to be digested is excreted in the form of feces from your body. This is the cut section or cross-sectional view of the gastrointestinal wall, which shows that the innermost layer is the epithelium surrounded by the mucosa. Then you have the submucosa and the circular muscle layer, which is basically the horizontal muscles that extend around the gut, and you have these longitudinal muscles, which are the vertical muscles, you can say, that extend longitudinally through the gut. And there you can see the mesenteric plexus, which is present in the submucosa and the circular muscle layer. Then you have this outer layer, which is the serosa. So your smooth muscle cells are connected to one another through these junctions, which are called gap junctions. These junctions are responsible for travel of ions from one cell to another. So what happens is, if your one cell gets excited or changes its membrane potential, these ions also diffuse to the other cells because they are connected at some point. So as a result, the whole muscle layer gets excited and causes contraction. This togetherness is called syncytium. There are two types of waves in your gastrointestinal wall: the slow waves and spike waves. Slow waves are basically the undulating changes in the resting membrane potential, and the spike waves are basically the true action potentials that are generated from the slow waves and are responsible for muscle contractions. So slow waves are produced 3 per minute in the stomach, 12 per minute in the duodenum, and 8 or 9 per minute in the terminal ileum. So how these are generated? These are the interstitial cells of Kajal, which are also called the pacemaker cells of the gut. These cells have sodium channels which open and close periodically and allow the ions to move in. When these ions move in, they produce an electrical ionic gradient, which is responsible for the production of electronic gradient, which is then transferred to the smooth muscle cells. These smooth muscle cells make these ionic gradient to travel from one cell to another. As a result, the whole muscle layer produces slow waves, spike waves. So what happens is that normally your resting membrane potential is of a cell, intestinal cell, is -60 or -50 millivolts. This resting membrane potential is changed due to slow waves due to the influx of sodium ions. These sodium ions slowly comes into your cell, and when they changes the resting membrane potential from -50 or 60 to -40, this is the time when these calcium sodium channels, which were inactivated before, gets activated, and they cause the sudden influx of the calcium and sodium ions, which is responsible for the action potential or spike potential, which leads to muscle contraction. Then you have this hyperpolarization and depolarization state. The hyperpolarization state is the one in which the cell is resting membrane potential is much more negative than 60 or 50, and depolarization is when the resting membrane potential is less negative than 60 or 50, like 40. Risk factors that cause hyperpolarization are epinephrine, norepinephrine, and stimulation of sympathetic nerves, which also release norepinephrine. And depolarization is due to stretching of the muscles, stimulation by acetylcholine, or stimulation by GI hormones. Tonic contractions. These are basically contractions produced by some smooth muscle cells, which are continuous contractions not associated with slow waves, and these are produced due to continuous or repetitive spike potentials or hormones, or continuous influx of calcium ions. Quick test. What is called the true action potential? Slow wave potential, spike wave potential, or tonic contractions? The answer is spike wave potential. And channels involved in generation of slow waves: sodium channels, calcium channels, and calcium sodium channels. The answer is sodium channels. Thank you.